Good morning, guys. So we have a new face in Joshua White's course. Now we have Stephen Beckett, and I will start his introduction. So it's my pleasure to introduce you, Stephen. Stephen has a bachelor degree in geography and mathematics by the University of Leeds. He got his master's degree in mathematics in the live environment from the University of York, and his PhD degree in biological sciences from the University of Exeter. Oh my God, that's really hard to say. I read and I say wrongly sometimes. He then became a postdoc researcher, and currently he's a research scientist at Georgia Tech Institute in Joshua's lab. Stephen is interested in improving understanding and representation of mortality process in marine microbial populations and in large ecosystem models. He's also interested in understanding how surface marine microbial communities organize around natural oscillations and cycles, and more recently, I guess, understanding how leveraging data can improve our understanding of pandemic-associated risks and help with decision-making and outbreak mitigation. So please help me to welcome Stephen Beckett.
Thank you, everybody. And one extra piece of information that Flavia missed is that we actually met during our PhDs about 10 years ago at a workshop. So workshops are fantastic. Yes, you same. But yeah, thank you for having me, and excited to start doing this lecture. So this is following on from the lectures you had from Jacopo Marchi the last few days. And now we're going to be moving on to thinking about predator-prey dynamics and evolutionary ecology. And then, just so you know, then this week we have 2 lectures, so today and tomorrow. Next week we'll be doing the labs, and that will be on Thursday and Friday after the break. And then in the week after that, we'll be moving on to thinking about outbreaks and epidemics. So that'll be a new topic. Okay, so I'm going to get started, and I'm going to start asking some questions. So question 1, what does it mean? What is evolution? Does anybody want to kind of throw in and give an answer to that? Go ahead.
For me, it's descendants with change.
Okay. Does anybody else want to throw something out there?
I guess it's like speciation and diversification of organisms.
That's good. Anybody else? I'll ask one more. Yes?
I'd say it's the accumulation of genetic variants in a population.
And I think everybody is right. So I'm just trying to get you interacting and talking with me. The definition we have here is thinking about genetic change and the change in genetic makeup of individuals in populations going through subsequent generations. And then what is evolution via natural selection? I'm going to throw it out there again. Can somebody raise their hand and try and answer this? Go ahead.
It's actually a question because
Sure.
Isn't evolution like necessarily by natural selection or artificial selection? No? I thought it was.
So
okay.
Imagine that you are in a totally homogeneous, so very plain, very simple world. This is a simulated theoretical example. And so everything is the same always. And let's say we have some kind of microbe, some kind of bacteria in that population, and it just grows and grows and grows. Is it always going to be the same? Or is it going to change? And that kind of comes back to the question you asked me. And so there will be some changes in its genetic makeup just because of the randomness within the division process. But that evolution in that very plain world is going to be undirected. There's no selection from anything in that world, so it will be random mutations. So yeah, selection isn't always necessary. And so what do we mean by natural selection? I have a question.
I'd say, okay, that's easier. I'd say evolution via natural selection would be the success of a specific genetic characteristic that would establish that individual or that specific genetic makeup as the dominant one in its environment.
Okay. Does anybody else want to throw out an answer to this question? At the back.
I'm not a biologist, so just to be clear, maybe this is wrong. I just continue on Paula's line of thought. I wouldn't say dominant, I would say surviving, because I think that's the main point, like not dying. I don't know.
Yes, so selection is kind of, there are some things that are favored over others. And so if you run the clock forward through time, then those which are more successful against that pressure are probably going to grow more than those who are not. So you're going to change, and to bring it back to the textbook answer, we have any non-random changes in the genetic makeup of individuals due to differential reproduction of individuals. So those individuals who are favored due to the environment are going to reproduce more, and then those individuals are going to make up more of the population, and so those individuals are then more likely to reproduce and become the more dominant members of that community over time. And final question before we move forward, and that is, what is ecology? And again, I'm going to ask for answers from the class. Nobody wants to answer? Oh, go ahead.
I'd say it's the study of interactions between organisms.
I would agree with that entirely, and but there are other definitions too, and certainly where it appeared from, it was thinking more about natural history and how organisms survive, and so it's actually slightly broader. It's those interactions between individuals, but also in the context of the environment that they're in. But I like to think about it more as the individual interactions between different species, and certainly there are fields like sociology who also use these kind of approaches, which are essentially these interactions between individuals and how they might behave, and it's kind of the basis of thinking about social network behavior from my understanding, though it's probably not how they arrived at it. And so, yeah, organisms interact, but they're embedded within these much more complicated food webs and structures, and there's different environments that you can flow between, and so there are these very complex networks of interactions between different organisms and environments in which they live. And so, one more question. I forgot about this one. What does it mean to say evolutionary ecology? So merging these kind of processes together, evolution and the ecology. And it's essentially, merging those two concepts. So the study of effects of evolutionary change on the interactions amongst organisms and their environments. And in the past, this has mostly been thinking about phenotypes. And what a phenotype is kind of the observed changes and features of specific organisms. So what we can see and measure through experiments. It's only very recently that people have started looking at the genetic level of these kind of organisms and their interactions, and trying to understand the molecular biology of these kind of interactions. And so, going back to these interactions, organisms can interact in multiple different ways. And so we have a kind of predator-prey situation here in the top left, so hunting and between organisms, so the predator gains, but the prey loses. We have mutualisms, so this is a plant pollinator system. The bees are pollinating the flowers, the bees get the nectar, so they both win, and they both get something from this interaction. We have a competition between trees for light, and so you might get some shading of the taller plants over the ones lower in the canopy, and so that's kind of a loss for those at the bottom, where the top isn't really gaining anything from this interaction, it's just happening. And then there are other interactions as well, such as using resources and recycling networks. And so, yeah, we're here at a quantitative ecology workshop, so we want to ask questions like this. How can we use models to try and make sense of all of the biodiversity that we see out there in the world and across different scales? So we can think about interactions at the microbial level and think really, really small, thinking about bacteria, amoeba, viruses. Or we can think a little bit bigger. So here I'm showing various diatoms in the ocean, which are quite a lot bigger than bacteria, but still very small. But we can take these models all the way up to thinking about rainforests and ecosystems as well. And so different researchers will have different questions, and they'll want to use different models to try and assess what they want to get out of it. But at their basis, a lot of these models are very similar, and they're addressing very basic interactions. But yeah, to get towards these questions, we need to really simplify and drill down. And so theoretical models in ecology have been around for quite a long time. And I'm going to talk about the Lotka-Volterra model in a second, which was independently brought up by these two individuals, Vito Volterra and Alfred Lotka, back at the late 1800s. And yeah, Vito Volterra, he had a son-in-law who worked in the fisheries in the Mediterranean, and he was really interested about why the fish stocks kind of changed year by year and season by season. And so Vito Volterra ended up studying this system and arrived at a set of equations to try and think about why this might be happening. And this is the model that they came up with, and Lotka came up with this same set of equations simultaneously. And so we have one equation, dx dt, which represents the change in a prey species through time, and another equation, dy dt, which represents the change in the predator population through time. And so here, we're saying that the change in the prey population is this xa minus bxy. And so the A represents kind of the growth and the birth of the prey species, whereas the bxy, that's talking about the death of the prey species due to the predator. And similarly, for the predator term, we have minus cy, so that's a death of the predator, and a plus dxy. So that's a gain for the predator from consuming the prey. And the reason that the D and the B are different, and you might have seen this in other lectures on this workshop already, but the reason they're different is that there might be some efficiency associated with this interaction. So the predator doesn't gain all of the prey it consumes, or there's some conversion between prey into predator, which isn't a one-to-one mapping. And the results from studying the system give you these kind of limit cycles in the phase space between the x and y. So if we plot the dynamics of x and y through time, depending on where you start, you get these kind of oscillations. And this very simple set of equations is essentially the basis of what most people in theoretical ecology still use today, regardless of the system that they're looking at. So this is yeah, a very important equation as far as we're concerned. And so, yeah, this coupled pair of ODEs that we've looked at a second ago, if we look at it through time rather than the phase space, we see these kind of marked oscillations. And so there are some important features here, and you'll see that the prey and the predator do not peak at the same time. And so the important kind of reading here is that when the prey increases, there's more prey available for the predators to find and catch and consume. And so the predators increase after that increase in prey. But when the prey are very abundant, then the predators are catching a lot of those prey, and so they go down. But when there are few prey available, then the predators aren't finding enough to sustain their population, and they go down. And so we get these on-off cycles. So the prey rise, and then the predator rises, the prey goes down, and the predator goes down. And this is kind of an important feature that I want you to keep in mind as we go forward. And yeah, here's the phase space again. And I'm showing null lines and trajectories in this space. I don't know if you've come across those terms in what you've learned so far. Could you go ahead and tell us what the null lines are? Oh, we got one.
There are lines where
That's right. Yes. I think everybody's in the room today, but yeah, we'll repeat.
All right. So there are lines where the derivative of the change in the population is zero. One
That's
of the
right.
populations doesn't change.
Yes. And then the trajectories are what we're showing in the black curve. And then which way round do these trajectories go? Do they go around this way or around that way? Counterclockwise, yes. So they go around this way. So the prey goes up, and then the predator goes up. The prey goes down, and the predator goes down. And it goes around and around and around. And we have this kind of equation that describes this. It's not true limit cycles. And then does this kind of exist in the real world? And so this is also a very famous kind of data set describing the lynx and hares in Canada, the Hudson Bay Company. And yeah, these were hunters going out and finding furs of these various species. And so the number of those changes year by year, and you see that there are also these kind of oscillations associated with this data set. And it isn't so nice as what we saw in the model. Models are very simplistic, and they can be an idealized point in the world, but the real world can be much more complicated sometimes. But some parts of this graph we can see are a little more what we'd expect towards the end, whereas the beginnings may be a little more random and chaotic. But here, at least after 1905, we see the prey go up, and then the predator go up, and the prey go down, and the predator go down, which is what we expect from this simple model. So now I'm going to talk about a slightly different system and start thinking about competition between individuals. And so this is an experiment by Gauss in 1934, and he did an experimental study of these rotifers called Paramecium, and they're in a fixed environment. And so what I'm showing in this graph here is two species of Paramecium, so Paramecium aurelia and Paramecium caudatum. And the two lines in each of these graphs correspond to two different experiments. So the top line in both of these panels considers the growth of these species when they're grown alone. So we see the abundance go up and then kind of saturate in both cases. And the second line is when they're grown together. And when they're grown together, we see that the abundances don't go as high as they would when they're grown alone. And even for Paramecium caudatum, it seems to decline through time as well after it reaches its peak. So there's some competition here between these different species. And the reason for that is probably that they're using the same resources to grow. And Gauss kind of categorized this with this title, The Struggle for Existence, which is very dramatic. It captures your attention. And so here on the right, it's these top two images here, which are actually these species that Gauss grew in these experiments. And yeah, what I just said, these species are probably competing on the same resources to grow. And so if we consider that each of these species could be called N1 and N2, then we can write down some kind of theory which says that in order to get new individuals, we must consume resources. And so if N1 consumes a certain amount of the resource R, then we can divide and get 2 lots of N1, and the same for N2. And moving forwards and writing that in an ODE, we can write down some equations which look a little like this. And so we have the change in resource, which in this case, there's going to be some kind of flow through this environment, which I'm modeling through this term omega. And so this is saying we're adding resources all the time, but they're also getting washed out of the system. And then the resources also disappearing due to uptake by each of these species. And these species have some kind of conversion based on what they can uptake to turn that into their own individual matter. And there's some death of these individuals as well. And we can simplify the system a little bit if we assume that the resource is kind of taken up fast. So we can say that dr dt is essentially 0, so the resources being added are essentially being consumed straight away by these individuals, which allows us to set dr dt to 0. If we then reorganize this kind of set of equations, we can find R, which we'll call R star, the steady state of the resource through time, which is given by this equation. And if we plug R star instead of R into this equation, we can split this down into a much simpler set of equations, where there's just 2 equations now rather than 3. And you'll see that it's exactly what we had before, but instead of R, we now have R star, which is dependent on the abundances of N1 and N2. And so if we substitute that back into the equations, then we get this kind of horrible looking thing. But this is quite similar in some sense to what we saw before. And so I'll just do the prey equation that we saw previously, which was something like this. But I'm going to write this a little bit differently, because in this case, we're thinking about a situation where there isn't a predation term. So we could have something like ax minus mx. And I'm going to step again to make this more complicated, or to get closer to this set of equations, by saying that this term isn't exactly what this term says. And in this case, if we just had ax, then you're going to be in a position where you're describing exponential growth. So there's no kind of competition here. And so I'm going to move to what is the logistical equation within the Lotka-Volterra framework, which would be something which looks like this. And so I don't know if you've seen this equation before. You have? Great. So what does K represent? That's right, yes. So that's the carrying capacity, how much of X you can have in the population. I hope you can kind of see, if you look back at these equations, these are somewhat resemblant. And so in the equations on the slides, then we have A, which is represented by epsilon, gamma, R naught. And then we have this K term, which is this latter equation, which is some dependency on both of those individuals, which represents this kind of K. So if we were to rewrite this for two species, we might have something like, and I didn't draw this in the right place. Let me start again over here. So in this case, the carrying capacity is for X and Y. And that's essentially what we're doing here. Yes?
You assumed that resources were stable, so everything that was coming in was consumed. And then you put the R star on the equation and assumed also fast dynamics, you said. I didn't understand why that's fast dynamics, why omega bigger than gamma n represents a fast dynamics. And
sure, so, yeah. So, yeah, we're adding in these resources as fast as they're kind of consumed. And so the level is going to be constant in that case, but it's going to it's still dependent on what N1 and N2 are. So there's some dependence still built in there. And if you add more N1 and N2, then your R is going to decrease. But this is kind of an approximation of that larger system that we saw in the first place. Does that help?
Yeah, it does. Thank you very much.
Great. And so, yeah, this is just the Lotka-Volterra model, but with competition added. And so this is a way of implicitly representing resource dynamics. But in this case, in this system, only one of these species can survive. And so we have this kind of mathematical expression which comes out of this system where if M1 over epsilon gamma is less than M2 over epsilon gamma, then N1 survives. But if that is not the case, then N2 survives. And so I can ask, what does this term represent? What does M1 over epsilon gamma mean? And does anybody want to have a go at that?
Is the ratio between the mortality and the birth rate?
Exactly, yes. So if you're dying faster than you can grow, then you're not going to stick around as long as the other person. And so this is the kind of the basis for resource ratio theory, which came up with in the 70s and 80s by David Tillman and others. And he said, if multiple species are competing for a single limiting resource, then whichever species can survive at the lowest equilibrium resource level, i.e. the lowest R star, they can compete all other species. And so I'm going to show kind of an example of that now. And so we're going to write up another set of equations, and here it's quite similar to what we just saw, but I've modified what the consumption of resources looks like a little bit. So in this case, we have this kind of saturating term given by R plus K, and this is what is called the monod growth function. And so the idea is that you can only grow so fast, and so if you have unlimited resources, then you can only get up to a certain growth rate. So you're going to have a saturating growth response with more resources on the x-axis. And so there's a nice feature about this set of equations as well. And here K doesn't represent carrying capacity. It represents the half saturation constant. And so when R equals K, then the growth rate will be at half its maximum level given by mu. And so that kind of sets how steep and sharp this curve saturates. And so I'm going to move forward. And now we had this simple model. I'm going to extend it to think about multiple different populations. And they're all given by these subscripts I. And so it's exactly the same model that we had before, apart from now the resource is being consumed by all of those different species. And this is, in the bottom right, is a simulation in which I'm adding a new individual to this environment every 10,000 hours. So we have the purple, which is added at a very small amount. It grows up to some saturating level. I add a red species here, which you might only just be able to see, but it does not survive. It is outcompeted by the purple species. But then at time 20,000 hours, the blue species comes in, it outcompetes the purple and becomes dominant in that environment. And then there's a few more dynamics after that. And a way we can try and think and explain why this system looks the way it does, you can go back to this R-star theory. And so if we take this very simple kind of model on the left, we can rewrite that to find R-star. And it has a slightly different form than before because we have a different set of model equations. And we can rewrite that to think about mu as a function of k, because I think this kind of helps explore why the simulation looks the way it does, which is given by these kinds of lines here on the left panel. So on the y-axis is mu, the growth rate, and on the x-axis is k. And the colored lines correspond between the two panels, where a higher R star is in this kind of bottom right corner, and the lower R star is in the upper right left corner. And so we started on this kind of purple curve, and we tried to add the red species, which is on this curve with a higher R star. And so that couldn't invade because it's the species with the lowest R star which can outcompete the others. And then the blue curve comes in, and that is, where is the blue curve? It's here, the same place as this yellow curve. And so the blue curve is slightly higher with a lower R star, and so it can outcompete the purple. And then at time 30,000 hours, I'm introducing this yellow species at a low abundance. And the blue and the yellow are in exactly the same trajectory. They're on exactly the same function. They have the same R star. And so that yellow species comes in, but it can't outcompete the blue species, so it just sits there at this low abundance. And then we move forward to this kind of brown curve and the green curve, which I brought in at the same time at 40,000 hours, and they both have a lower R star, so they both come in, but they grow to different saturating abundances, and that's because they sit in slightly different places along this mu k distribution. And so they have different saturating abundances, but they can both coexist in this environment. And then finally, I'm adding in at 50,000 hours this top line, which has the lowest R star of all of these species and outcompetes all of the rest of the system. And so to kind of summarize everything so far, we're thinking about the changes in ecological dynamics, and alone they can predict the change in abundances of population through time. And this is all in a situation where we have fixed traits. And going forwards, we're going to think about how we can apply these in microbial systems too. Well, there are predator-like viruses. And so just to give you a kind of summary of what the biology of this kind of system is we're thinking about bacteria, which are these big blue blobs. And these little things here are phages, bacteriophages, or viruses of bacteria. And so we're thinking about these viruses attaching, absorbing to the host cell, injecting their genetic material, which is then replicated within the cell, and creates these new viral particles, which can then assemble. And at some later time, they kind of burst open these microbial cells in an event we call lysis. And they're released into the environment, and they can go out and find new hosts. And so these are an obligate parasite, but it functions quite similarly to how a predator-prey system would work. And so one set of equations we can write down to kind of describe this model is given here, where we have R as our resource, N as our bacterial host, and V as our virus population. And so there are a bunch of interactions here, like we've seen in the other models. So we have resource inflow-outflow, growth of the bacteria on the resource. We have this kind of consumption term by the viruses and some loss rate. And then the viruses, well, they're inside the cell, they're producing a bunch of new viruses, beta new viruses, before they lyse out of their cells. But then they're also attaching to host cells, so there's some loss there too. And so if we kind of run this kind of situation, this model, we end up with getting these kind of saturating limit cycles. So we see some predator-prey type oscillations, but then they saturate. And this is a paper by, yeah, go ahead.
I was just wondering why the mortality rate for the bacteria and the virus is the same as the inflow.
Oh, that's still here. Yes, good question. And so I've changed the environment a little bit to what we saw before. And in this case, we're also thinking about these virus and bacteria exiting the system because of this flow. And we're saying that that flow is faster than the individual biological processes of mortality. And so, yeah, if you wanted to write down all the mechanisms, then, yeah, you would want an additional term representing mortality. Okay. And so if we go back to the phase plane for this model, we see, again, these kind of counterclockwise cycles. So high prey means the predator, the viruses in this case, can grow, then the bacteria go down, and so on and so forth, but in this kind of limit cycle system in this case. And we can kind of modify this model. So we had explicit resources, and now we're going to an implicit resource model. So we're getting rid of the resources, and we're using this kind of logistic term. And in this case, we've actually added an extra equation to represent infected cells. So we've added an extra compartment in the system. So cells before were just bursting open, but here we're explicitly representing that middle step where viruses are inside and propagating within their host cells before they burst out. And so here we have this eta I part, which was different from before, and this is representing the length of time those viruses are sticking around before they burst open and kill the cell. And again, in this representation, we see this counterclockwise cycles. And there's another way of writing down this kind of way of trying to represent this time period that you're spending in this infected state. So you could kind of explicitly represent a delay by using exponentials. And so these two systems are effectively doing the same thing, but they're articulated in a slightly different manner. And they both end up with the same kind of features. And so what do we actually see in, yes, go ahead.
Can you go back to the slide with the lytic cycle of the phage?
This one?
No.
This one?
No.
Keep going back.
The one with the phages and the yes. So this represents the lytic cycle where the cell outbursts, but there's also the cycle where it does not do that and it implements the genetic information and now it's part of the cell. My question is, can we model that and like we do with this one?
So yes, we can, and that's called the lysogenic cycle. And so in that system, as you said, the viruses are integrating their genetic material into their hosts, and then those viruses are being propagated through the environment because those host cells with the virus material inside are dividing. And so that's a different way of those viruses propagating through their environment. They move with the host rather than necessarily lysing straight away. There's a lot more different infection strategies as well. Some viruses use a temperate strategy where sometimes they follow this lytic cycle and sometimes they follow the lysogenic cycle, for example. So there's a whole bunch of different viral lifestyles.
But that would be a different model, or can we do the both in a single model?
So I think it depends a little bit on the system, and certainly these equations that I've written down here are explicitly for the lytic cycle. If we wanted to have the lysogenic cycle, then we would want to have that infected state, so we'd want models similar to this, but we'd probably also want those infected cells to divide. So that would be the simplest way forward, but you might want to add extra mechanisms in there too. But yes, that's a great point. And yeah, this is one lifestyle of viruses that I'm looking at today, but others do exist as well. And different systems follow different rules. And so this is one example of data for a virus bacteria system for T4 phages and E. coli in this paper, Bohannon and Lenski. And we see that we do see these kind of predator-prey type oscillations, at least in this first 200 hours of this experiment. But if we then kind of move forward through time and we look at the whole time series here, going up to 600 hours, then we see something kind of happens in the system at around 200 hours. Does anyone want to kind of speculate on why the host might saturate after 200 hours, but the virus kind of continues to oscillate? Something in the system has changed. Yes.
The host developed some kind of resistance to the virus and managed to reach their own carrying capacity.
That is exactly right. But there's actually a little bit extra on top too, and that is that some of the hosts gain resistance, but not everyone, at least within this time window. And so those resistant hosts, as you say, are able to get towards their carrying capacity, but the susceptible hosts, they're still around and the virus is consuming them, so we end up with these same predator-prey dynamics, just at a slightly different level than we had before. And this kind of feature isn't only limited to these kind of virus systems. Here's a different paper in 2014, and the data I'm showing here on the right is from an algal rotifer type system with bacteria, so it's E. coli again, but now they're being consumed by this rotifer called D. discodium. And so similarly, we kind of see where in the first period, the dynamics kind of act in this kind of predator-prey cycle type way, but at some point, something in the system changes, which leads to then changes in the population dynamics that we observe. And we can see this in the phase plane too, that the first period has these kind of cycles, but then we get some different type of cycles after this kind of evolution feature has occurred. And they found in all of the kind of meta-analysis that they did of the literature that about half of the population timescale studies that they looked at had these kind of eco-evolutionary cycles going on, where you had this kind of disruption, these more cryptic population cycles going on in the phase plane. And so I want to go back to virus microbe systems here and kind of think about the co-evolutionary dynamics between viruses and hosts. And so, yeah, viruses have a very strong selection pressure on their hosts, and they typically absorb to their hosts via receptors on the cell wall. And there are various different resistant mechanisms that the bacteria can exhibit to try and prevent viral absorption or viral replication within the cell. So there's both extracellular type defenses the bacteria can use by changing what the receptors on the outside look like, for example, but they also have intracellular defenses as well, where on a genetic basis they can kind of prevent the viruses from replicating. And so here we're going to ask about how the populations of these viruses and hosts change through time when there are multiple genotypes within a particular population. And we're going to look at this kind of system where we have 2 bacterial species and 2 phages. So they're the same kind of species, but different strains within this kind of system. But then there are asymmetrical interactions. So this top phage has very strong interaction with the blue bacteria, but much weaker with the green, whereas the purple phage has kind of weak interactions with both. And so in this system, you see we get cycles in the total virus and the total host. But if we look at the sub-strain level, then we get all sorts of extra interesting oscillations, which sum together to something nice, but are individually quite complex. And so if we look at the total host and the total virus, then we get this kind of oscillation. And is there something different about this oscillation than what we've seen before in the previous models? Yes. Here, the phase plane, things are going clockwise. And this is due to what is going on at the subgenotype level. Hey? Okay. No, that's good. You're okay. You're okay. And so looking down at the genotype level then, we have this kind of blue bar, which is representing the bacteria genotypes. So the filled area are the bacteria with low vulnerability, whereas the white area is bacteria with high vulnerability to phage. And on the opposite side, this red bar is going to represent the genotype frequencies within the virus population. So those with low offense versus those with high offense. So for example, in this particular case, there are more bacteria which are highly vulnerable and susceptible to being attacked by phage. And at the same time, the phage that are there are mostly of this kind of lower offensive strategy. And so we can see within this phase plane, oh, I went the wrong way around, that the genotype frequencies actually change along this phase plane cycle. And so if we start at a particular position, let's say at the top, then here we have mostly vulnerable bacteria and mostly low offense phage. And so this is a position where the bacteria are in a position where they can grow. And so they grow, and the low vulnerability type is able to gain more standstead in this population, whilst the phage with low offense continue to dominate. But as we get more and more bacteria which are highly susceptible or low vulnerability, then the phage with the higher offensive trait are able to gain traction. And so we end up with this kind of cycle which goes in a whole different way round than what we expected in the case when there wasn't evolution at play. Yes.
What is in my mind right now? Every time you have a cycle, you have a new mutant, or do you have the same mutant shifting strategies?
So there's no new mutants coming into play. It's more the changes across this kind of genotype space are changing. So you either have, of your two strategies, you either have one strategy with more or the other strategy with more. But that kind of, how well they do, the selection pressure is changes as you go through this kind of cycle.
Yeah, because there are two
strains.
Yeah, two strains
yes.
here. Okay, I just wanted to make sure I understood. But would it be possible for viruses to like change a strategy rather than the genotype? Is that something possible?
So it could be possible. I don't think we're actually building that into this model at this stage, but you could certainly assume that there's some probability of them changing their strategy or moving from the low offensive strategy to the high offensive strategy, or from the high vulnerability strategy to the low vulnerability strategy with some kind of mutation probability. So that's a possibility. In this case, I don't think we build that into this particular model.
Okay, thank you.
So I was just wondering why the low offense virus has fares better against a low vulnerability bacteria.
So I think this is partly that there might be another trade-off going on that I've forgotten about in terms of growth rate. And so I think if we go back here, then I think the blue bacteria in this case, whilst it is highly susceptible to the phage, or it has low vulnerability, I think this type can grow a little bit faster than this green type. But where the green type has higher defense or is less vulnerable to the phage attack. Are there any more questions here? Because I know this part is not simple. Even I'm struggling a little to challenge. Okay. And yeah, these slides are all up on the Google Drive as well. But yeah, so here we get clockwise cycles, and that's because we have these changes in the genotype frequency, which then mean that if you're looking at the total population level, things work a little bit different than in our previous models where we weren't thinking about evolution. And so we can kind of ask, does this happen in real-world data sets? And in this case, we're going to look at a chemostat system, which is kind of similar to what I answered before in terms of this flow through the system. So the resources are coming in, and then they're going out. The bacteria are being washing out, and the viruses are being washed out as well. There's a flow through the system. And in this data set, we see there's some initial kind of dynamics before it settles down to some kind of oscillations. In this kind of time series sense, it's quite often hard to see what those oscillations look like. But we can kind of identify some areas where it looks clean, whereas others maybe not so clean. And if we go into the phase space and look at these different time periods, then we see that things are moving clockwise, which is further evidence in this case that we're probably looking at multiple genotypes underneath these kind of species level claims. And so, yeah, to kind of summarize some of these ideas, then when we don't have evolution built in, then we see these nice counterclockwise cycles where the prey peaks first and then the predator peaks after that. But when evolution comes into the picture, things look a little bit different, and we can get cycles which go clockwise, where the peaks and troughs can even align in some cases, and we get different kind of lag in the system. And yeah, if we go to full coevolution, then we really get these kind of clockwise cycles. I did it again. I'm too used to doing the counterclockwise. So yeah, on the left, no evolution. On the middle is evolution of one side of that interaction, and on the right is evolution of both sides of that interaction. And to kind of highlight these dynamics again, we have a canonical, theoretical predator-prey cycles. If we build in evolution on one side, so for example, a host becoming resistant to the phage, we can end up with these kind of cryptic dynamics, which look very difficult to explain in what we measure. And if we end up with a co-evolutionary system, so both the virus and the hosts are co-evolving, then we can get these really interesting features where we might get these clockwise cycles appearing. And so I kind of want to leave here thinking about this question, and that is, if competition is prevalent, then why do we actually see so much biodiversity in the real world? These seem to be kind of very counter ideas. And yeah, if everything is going down to this low R star where you're getting out competition, these kind of simple experiments, why in the real world is there so much biodiversity? Why do we have multiple species coexisting with one another? And so this kind of issue is known in some circles as the paradox of the plankton. And it's this idea that you get lots and lots of biodiversity, lots of different species coexisting, but that's not what you'd expect from these kind of simple ecological models where you expect the individual that consumes the resource the best to outcompete everything else. And this kind of issue is still being discussed today. It's an ongoing kind of question. People have posited lots of ideas about why that might be the case, but it's not a fully resolved question. I don't know if anybody wants to kind of think about why things might be different in the real world than in these kind of experimental systems. What might be leading to multiple species coexisting together? What are the assumptions which are different between those systems? Go ahead.
So our models always assume heterogeneity between environment and also biotic factors. Something that we were looking at class with Prianga is that heterogeneity can lead to biodiversity, both in spatial factors, temporal aspects, and as well as biotic different pressures for different predators in different regions.
Yeah, that's a great point. So yeah, different areas will have different pressures acting within them, which may provide a benefit to one species over others. And if that changes across space and time, then you might have pockets which survive of different species. Go ahead.
We have also looked into a bunch of different systems where, for instance, we saw one resource competition and one in the two species, one predates the other, and then there's a bunch of processes that actually, with the right conditions, allow coexistence, but they're not being considered in the traditional Lotka-Volterra model. So
definitely, yes. And in the back.
And I also think about timescales. I still couldn't grasp exactly where it fits on all of this that we're doing, but I mean, we've seen transient phases, and we've seen that most of the time when we analyze a model, we consider it at equilibrium and say, oh, that's what's going to happen. But in the real world, you just have many systems and they interact with each other. There is no closed system. Like, yeah, it's kind of in the line with what other people said too, but that's what came into my mind.
Yes.
There is also the interspecific trait variation that maybe can act in individuals.
That's right, yeah. And definitely within a population, you're going to have some variation too, which might favor different parts of that population depending on which environment you're in. So, yeah, biodiversity isn't just different species necessarily. It could also be different strains within the same kind of species. That's another important factor to consider, yes. Does anybody else have any other ideas? Yes.
I would also consider that like having multiple species creates new opportunities for many other species. In the case of like the number of species or the abundance of a species changing the environment and creating microclimates and new niches for other species.
So feedbacks between species and the environment, that's a very good point, yes. Or even resource recycling, for example. So if some bacteria are eating some compounds, and in the process of eating those, they produce some other compounds, some other bacteria might be able to eat that. And so you can get facilitation of existence through that method. Yeah.
Something else that we also looked into is stochasticity and how it can play a role in helping systems that would be on a fixed point to keep on oscillating, and that could also help other species.
Have to find some new batteries for that one, yes.
That was the main point, stochasticity, and how different species can have an advantage when there's an abundance of resource and the resource is scarce, and stochasticity, when added to a system, can create those variations that could lead to more favorable outcomes in biodiversity.
Yes, go ahead.
I believe there's also the effect of inter-competition to talk about. Like if you have a single species that competes too much between themselves and less with other species, that creates an opportunity for another species to grow. So there's that too.
So opening up a niche where somebody might come in and take what available resources are there. Yes, that's definitely an interesting feature too. And yeah, I hope this kind of, do you want to have, no? Okay, sorry. I saw the hand. Yeah, so this question potentially has a whole lot of different answers. These systems, as they grow, get more and more complex, and there are multiple hypotheses about why you might get diversity, and it probably differs depending on which system you're looking at to what might be the dominant factor, which makes it challenging to come up with one unifying theory to explain this, which is why it's still being discussed in the literature today. I think all the points that you made are part of those underlying reasons to why we might expect biodiversity to exist and propagate. And there's probably even a few more that we missed. And I think this might be the end of my slides. Yes. Yes, we made it to the end. Okay. So I think we still have a little bit of time. So does anybody have any questions? Yes.
Are there any points that you would add to to the discussion?
Sure. A lot of my research involves thinking about microbes in the ocean. And we actually had a paper out last year trying to think about kind of microbial diversity in the North Pacific Ocean. So that's kind of near to Hawaii. And in that kind of system, we were looking at abundances, but also metagenomics and transcriptomic profiles. And there we were kind of looking over day-to-day cycles, so night and day. And yeah, in the ocean, phytoplankton are kind of the dominant kind of species in the surface ocean. So there's a strong selective growth pressure there between it being day and night. You can grow when there's sunlight, and when it's dark, you're respiring and dividing. And we actually found that different species there were adapting different strategies to uptaking resources from the environment. And different species seem to be uptaking nutrients at different times through the day, which was kind of very exciting to see. So that's what we term temporal niche partitioning. So going back to the niche idea, and because of these kind of different biological mechanisms going on between when you're trying to metabolize energy versus when you're respiring and dividing, then we ended up seeing this kind of chain of events where the phytoplankton and autotrophs were uptaking nutrients at a different time of day than some of the more heterotrophic species, so those which don't need sunlight as part of their energy. So that's one thing I could talk about. And I think one thing that we didn't cover here is kind of the diversity of interrelationships and interactions as well. Going back to this kind of resource facilitation, recycling type of idea, some of these interactions can actually help facilitate and sustain populations. And so you can think about these chains of interactions and interdependencies which can facilitate biodiversity. And so I guess going back to more ecology literature, you kind of think about these things called keystone species. And so the canonical example there is thinking about kelp forests and otters, I think it is, off the Californian coast. And I've forgotten quite how it works, but the otters are essentially consuming the kelp, which prevents the kelp from kind of taking over this environment and deoxygenizing the water. And if there's no oxygen in the water, then it's very hard for other species to survive. So by the otters being there, they're actually facilitating a lot of biodiversity in those systems. 2 more questions.
Since you No, it's okay, I guess. Since you talked about diversity of types of interactions, could you talk a bit about mutualism and cooperation and its role in creating biodiversity?
It definitely does. And yeah, plants and pollinators is the most easy to describe example there. So these plants, they produce nectar, which brings in these pollinators. The pollinators then take the pollen with them and visit other plants. And so they're able to help propagate plants across systems, but also sustain biodiversity in the system because they don't necessarily go just next door. They might go quite a long way. And so you help sustain the genetic biodiversity in those populations too, in those systems.
But like, do you have any example of that in your work with microbiota?
That's a good question. And I don't know about necessarily mutualisms, but certainly things do get a little bit complicated. And going back to this kind of lysogeny idea that was mentioned earlier, sometimes viruses can actually carry genes with them which could change the metabolism of the host cell when it becomes infected. And that's probably not a mutualism because it's for the virus's benefit in terms of helping it propagate further. But I kind of find that as a very interesting type of interaction where the virus can actually change the potential of that host bacterial cell to metabolize and live in its environment. But mutualisms, I think we have this kind of resource recycling idea which kind of could serve as a mutualism in some situations, but I'm struggling to think about if there's a direct mutualism, i.e. species A creates compounds that species B needs, and species B provides compounds that species A needs. I can't think of any examples of that off the top of my head. It might exist. It's more likely to be indirect, so maybe there's a couple more species in that link.
It's actually related about the course in general. I wanted to ask you if you could elaborate a little bit on how this course is going to go for us. You talked a little bit more about epidemiology, but are we going to do labs, and how's that going to be? It's a general question.
Sure, of course. So the labs will be a week today, and it's mostly going to focus on the ideas in this lecture. So thinking about predator-prey ecology and adding on some of the evolutionary links. And then also we'll be looking at an individual-based model in addition to some ODEs. And yeah, those are computer laboratories, so coding. Yes, yeah.
Going back to the mutualism subject, but still connecting with what Amanda asked, do we have models for mutualism or how they work? Is it something we're going to see in this course?
Not in this course, but I can find a paper and add it to the Google Drive. But yes, models do exist.
Okay, it's because there is a question that is in my mind, but I don't think how useful it will be to ask it now. It's because when I'm learning these models, they seem to be very exact in the concepts. For instance, you have here species 1, species 2, strain 1, strain 2, and then we biologists go to each other and ask what is a species, and there's still no definition established. And when I asked about intra-competition and interspecies competition, I was like, okay, if we don't know what a species is, how can we define if we have inter-competition or intra-competition in a model? So I wanted to ask, how does this affect maybe the building of models and such?
Definitely. Yeah, that's a great question, and part of why we are still building models, and part of why ecology and theoretical ecology isn't quite at the level of physics and theoretical physics. And a lot of that is due to limitations on what we can observe, and certainly going down to this kind of genetic basis has only been feasible in the last few decades. So it's very hard to exactly know all of the parts of the system. There's also this issue of scales, and so you might have things occurring at the molecular level, which then need to be translated up to the population level if you wanted to exactly nail all of the pieces. And nailing all of the pieces is really, really tricky. Some people try, and there's a whole literature of systems biology in which you're trying to think about how proteins might interact with one another and how that might scale up into metabolism and how that might scale up into changes in population abundance. but it's really, really hard. And so it's very often easier to approximate things in certain ways. How you do that approximation is also kind of tricky, but you have to do the best that you can with the data you have available and trying to make sure that the mechanisms that you're using are plausible. We had earlier this example where if we just had this equation, this would suggest that this population will grow exponentially, and we're pretty sure that that's not going to happen forever, so we needed a slightly different formulation. And this approximation might be useful in some circumstances. So if you were just to look very early in an experiment, then you might see exponential growth at the beginning. But later on, then that model wouldn't approximate the real world so well.
Thank you for the answer, and just give a conclusion to my thoughts. Yes, I would love an article talking about models in mutualism, because that's another subject that biologists go like, wait, it doesn't exist a plus without any kind of cost for one of the species. Mutualism doesn't exist, and that's a discussion that goes. So I would be very interested
definitely.
in how
yes.
mathematics and physics interpret that. Thank you.
Can we do this question first? Sorry.
So I was thinking about these cycles that you showed us today, and as far as I understood, these cycles that are more narrow.
The narrow ones?
Yes. They just happen when we are talking about these microorganisms like bacteria and stuff, because it's like, to me, it's related to the reproduction that is faster in these organisms, and I don't think I understood why some of them go on the other side, the clockwise cycles. So if you can talk more about this, and I also want to know if they only happen in these microorganisms as well.
So that's a great point, and microorganisms are kind of the canonical place we see this, but that's mostly because, as you said, replication and mutation are kind of linked, and the timescales here are very short. So these bacteria might be dividing in minutes or hours, which is very fast. And so if you have a mutation probability which is quite low, then you need a lot of replication cycles to happen in order to see mutation appear. And these bacterial systems, they have maybe hundreds of thousands of bacteria per milliliter. So there's a lot of bacteria which are dividing very quickly. So these are the kind of systems where it's possible to observe evolution in action, which might be a lot harder to do for more macroscopic organisms, where you might have to look at something like the fossil record to try and infer that genetic change was occurring. And it might be very tricky to then try and look at population cycles and those kind of features, because the ecology and the evolution would be happening on totally different timescales, because the evolution would be much slower than the ecology. And then, so in this middle panel, we're thinking about evolution of just the prey or just the predator, and that's kind of why it narrows in the way that it does. Whereas on the more clockwise cycles, then we're thinking about co-evolution. So both the predator and the prey are evolving, and the underlying genotypes are changing their dynamics as well, which leads to this opening up of that phase space and observing these clockwise cycles. Does that help? Of course. And yes.
You know what? I was just thinking about an example about what Laimara asked of mutualism, and I thought about transduction. That could be considered some kind of mutualism because now the bacteria is resistant and can live, and then the virus now has something to infect, so they both have a benefit. And I thought about that as an example.
Yeah, thank you for sharing. That's great. And then, did you have a question?
Related to your plankton research, so the plankton is like a basic resource in the food chain, and I would like to know, because there's a lot of interactions if you want to model the plankton, so I would like to know like which factors you have to include in a model related to plankton.
Sure. And in the research I discussed, we haven't actually tried to model that system yet, but in general, there are a suite of models which try and understand this kind of phytoplankton dynamics and where they are in the food chain. Most of them have a kind of key structuring trait, which is size. So the size of those phytoplankton is quite important in describing a lot of the other interactions which happen, whether that's encounters with different organisms, which is size-driven through diffusive properties at a very microbial scale, or more by how fast you can swim at slightly larger scales. But it also relates in not a linear manner, but there is a kind of relationship between size and how quickly you can grow and divide as well. So size is quite a key structure in a lot of these models. Some try, and certainly for the larger phytoplankton, they try and compartmentalize all of the different parts, but that means you can potentially have a really complicated model with hundreds of species, and if you want to move that from thinking about one ecosystem to all of the oceans, then it gets incredibly tricky. And so a lot of these models are trying to think about variation in traits, so these kind of growth mortality terms, and using size as kind of a key variable in terms of structuring on top of metabolism. So you might have a few different kind of life history strategies, and then embedded within those different strategies, you might have a continuum of size built in as well to try and represent biodiversity. But unfortunately, most of the models are not very simple, but people are modeling these systems, or trying to at least, yes. Okay, thank you all for paying attention and for asking questions and being involved and interacting. I'm going to wrap this up here. So I guess it's time for a break now. Okay, thank you.